Hello everyone, you are all welcome to lecture number 1119, and today's topic is interesting, and that is multicellular life evolves in the lab. This is the research paper published from Michigan State University, USA, America. And these scientists in state Michigan University, they are working since 1988. And that publication, their finding in the form of statistical data and experimental values and all the experimental findings are supported and it is quantified. So they have mentioned, actually in that paper they have mentioned that they have created life in the lab. But actually that life started from single cell. So that's unicellular. Cell was multiplied into multicellular. That is the biggest achievement, no doubt, because as per the opinion, I say opinion, because there is difference in opinion, forming the opinion by the scientist, and the scientific authentic reporting. Once we say scientific reporting means it is supported by experiment, statistical data, all instrumentation, and also it is quantified. If it is not having all these experimental values, then we will say it opinion. So there are a lot of opinion about the formation of or creation of life on this earth. The first opinion of the scientist is that when life was forming, there was lot of meteorite, asteroids, they were hitting the earth and they were having lot of amino acid molecules, and those amino acid molecules rain is responsible, and it was found in this deep space meteorite and asteroids. And once amino acid was formed there, then with the help of peptide bond, the simplest dipeptide and then tripeptide, polypeptide protein formation was there. So that is opinion. They don't have any such concrete experimental data to support this scientific statement, hypothesis or opinion. Now the question is, once we are talking of formation of single cell, if it is single cell, for example, and here there is no formation of any nucleus in the single cell in a very primitive stage, and then as you know that this single cell is having 42 million protein molecules, and according to the scientist's opinion, that it took 2.5 billion years to become multicellular, and from here multicellular it took another 600 million to become vertebrate. So this is what the explanation is given, that how this amoeba was formed. That was unicellular, that was amoeba. And this amoeba single cell, what it is mentioned, and it is found in marine colony of bacteria, amoeba. And then the USA, that is Michigan State University scientists, they found very common bacteria unicellular, that is called E. coli. This E. coli, they found that in 4 days' time, it is doubled. For example, if it was 1 million, in 4 days' time it will become 2 million. So they divided this E. coli in some 1 dozen beaker, and they allowed to grow. And in 60 days they found that it was having 74,000 generation. Were evolved. So within that few days time, this E. coli single unicellular bacteria was multiplied, and it has gone up to extent that it gave 74,000 generation. And to have 74 generation of multicellular like human being, we need at least 2 million years. And that was in 60 days. And for this evolution we need 2 million years. So it was found that this multiplication of unicellular in presence of good quantity of oxygen was having similar chemical properties and protein, amino acid, everything. Then they selected in their next experiment they started, that was in 2018. It was in 1988. They started with this, and then 2018 they started with yeast, unicellular. And this yeast, what they did, they supplied the nutrient, that peptine, that is a type of amino acid, dextrose, that is type of glucose. So nutrient and energy was supplied, and the oxygen was a little bit restricted, less oxygen was there. And it was found that within 60 days, there was lot of multiplication, and in that multiplication, with that restricted oxygen, some colony of multicellular was produced, was developed, was evolved, and they were separated. So from there, it was found that once the oxygen is controlled, that oxygen and pH value is very important factor. So this single cell, they become multicellular, and there will be some transition, adhesion of single cell and multi-cell, and thick film was formed. And even that thing is noticed today. That is called biofilm. That is called, and this biofilm is found in mucus, human mucus. This is a very interesting topic because of this multicellular discovery. They try to discover, that's why it is linked with this biofilm. Our biofilm is starting from our nose and it goes to trachea. And with this formation of biofilm, we call it biofilm, it's a thick layer, a sticky layer, which is found in our nose and it goes up to trachea. That is sticky, thick layer, that is called biofilm. And biofilm is nothing but it is the transition of unicellular pathogen bacteria which is entered there, and it is trying to convert, to multiply into multicellular. So there is transition between these two, and this unicellular transition to multicellular, it gives thick, we can say, mucus layer, and that thick mucus layer inside our nose and running to throat is called biofilm. And this biofilm formation is very, very dangerous. The reason is that this will not allow antibiotic to be effective, active. They will knock down. And secondly, this biofilm, formation of biofilm will not allow immune system cells to attack. They will be also clear. And today, the latest report is that there are up to 10 million people they die globally because of the formation of biofilm. So the biofilm chemistry of protein and transition or adhesion of unicellular and multicellular is a very complex study. In our COVID-19 period, in 19 and 20, that is 2019 and 2020, many people, they died because of the formation of biofilms, that mucus. So, and that is the concept of multicellular. So now it is clear that with controlled less oxygen, if nutrient is supplied, there will be formation of colony of unicellular to multicellular, and those multicellular, they have a tough membrane, that is cell membrane, and also there will be formation of nucleus with genetic material. So how the single cell became multicellular, and then multicellular again there was evolution and became multicellular invertebrate and vertebrate. And then evolution started first, once that single unicellular became multicellular, then lot of evolution was there and is going on still. Some are extinct, disappeared, and some are reappearing, new species. So this is what about the research paper, very interesting. It's really is a breakthrough news that they have already created the life inside the lab in the form of multicellular organism, starting from yeast cell, and that research they started in 2018. So here I have given about this life evolves in the lab, and that is multicellular. Today, Michigan State University, USA, could evolve in 600 days the unicellular into multicellular, and that they could not evolve, the nature, in 600 million years. They could do in 600 days. And this is most exciting experiment performed by this research finding. We will get the solution of mystery of evolution of life on Earth. This will give lot of information. And number two, in the year 2018, the same Michigan State University scientists that they grew fast with single cell yeast incubator, and selected those that grew faster. Actually, they first I have told you that they tried with that was in 1988 with E. coli, and then they shifted to, after getting lot of biological and chemical information from that colony I have already mentioned. Then they started with yeast shell, and that was in 2018. And this single yeast cell was put in incubator, and in that incubator and selected those that grew faster and they were bigger in size. They did this selection because they left the ones which were not big in size and healthy. And then they put in that nutrient and energy solution, that is peptone, amino acid, and dextrose, that is glucose, to provide the energy. And finally they found that the groups formed by the yeast cells were 20 times larger than their ancestors. And it is possible that was in 3000 generations that what they got 20 times. And with that thick formation of colony in millions, it was multicellular with genetic material with same properties. Number 3, the multicellular made of, that is eukaryotic cells. Eukaryotic cells means those cells which are having nucleus and genetic materials are present in the nucleus, have nucleus in which genetic material was separated. That is called eukaryotic cells, and these eukaryotic cells are actually multicellular cells, and they came from prokaryotic cells. Prokaryotic cells means they are single cell bacteria without the formation of nucleus, and they are very small also. Number 4, best example is given, that is formation of biofilms. That is, I have already told you, is very, very interesting chemistry and biology of biofilm. I have already told you lot of research is going on which are formed, and that biofilm is started from single cell bacteria into multicellular, which are sticky mucus blankets in our nose, goes up to throat, made by pathogenic bacteria in nose and throat to protect themselves. They are making such type of film. Those pathogenic bacteria, they were single cell, they are changed, multiplied into multicellular, and that is called biological film to protect themselves from any attack, either from antibiotics, what antibiotics is given, they will resist, and also from immune cells. So these two will not be effective, and that's why all will fail. And what it was happening during COVID-19 disease time, that was in 2019 and 2020, cells, and to protect themselves from antibiotic and immune system attack. It has 2 steps. This formation of biofilm has 2 steps. First step is that is your microbial adhesion, and that microbial adhesion is there first, and then second step is that is multicellular formation. So that is microbial adhesive and multicellular formation. They will be forming, and that is called biofilm. So here how this Michigan State University scientists they did this experiment, and it is very exciting research, and it has opened the door to understand lot of bacterial transformation, and they are evolving in multicellular and how they are making the biofilm. I have already told you. And also to understand how the life started on earth, and at the same time, if you are hearing my lecture, lot of investigation is going on by making the use of different tools of artificial intelligence. And today artificial intelligence has indicated, with the help of alpha fold software of Google, 200 million structures are indicated, and this is a big discovery. And once protein structures are discovered, because I have already mentioned in the beginning, even if it is single cell, in that single cell formation, there is involvement of 42 million protein molecules. So, and in that 42 million protein molecules, they are made up of the amino acid molecules by the formation of peptide bond. They are polypeptide. So, first question comes everywhere. People are giving their opinion. How the amino acid came, from where? That is the main, because amino acid is hooked to form the polypeptide, that is protein. And that opinion, I have already given a lot of scientists, they have a lot of different opinions. Some are giving their inorganic theory also, that some phosphate or some other sulfur and carbon, hydrogen, ammonia, they were all mixed up. But there are a lot of things which are only opinion, which are only hypothesis. But this research finding of that is Michigan State University of America is having all the experimental backing, all the statistical data, and it is authentic research finding. So with this I will stop. Thank you very much for watching the lecture.